In this lecture, we'll continue with chapter 7. Okay, so one thing that's maybe a little bit confusing is the difference between neurolemma and myelin. So shown here is a Schwann cell. So this is in the PNS, or peripheral nervous system. And you notice what the Schwann cell does is it will wrap many times around an axon. And that creates the myelin sheath, which you can see is a sort of dense layer near the axon. Let me see if I can point to it here. So this is the myelin sheath. And so it's many, many wrappings. Now, eventually, towards the outside of the cell, you have the nucleus and the various organelles. And this cytoplasmic area is called the neurolemma. It's the outermost nucleated cytoplasmic layer of the Schwann cell. And so it's important also to note that some Schwann cells will just have the neurolemma and not the myelin. So peripheral nerves that are unmyelinated will in fact have Schwann cells, just don't have the myelin, which is this wrapping. The peripheral nervous system has quite a good ability to regenerate. You could cut a whole nerve, and it's possible for the axons within the nerve to regenerate. And this is how the process works. Basically, when an axon is cut in the PNS, the severed part degenerates. So, for example, we can see here that an axon has been cut, so we can presume that the nerve has been cut or damaged. And so this axon degenerates. The distal portion of the neuron that actually touches the skeletal muscle fiber here would degenerate. But what's left over is a regeneration tube formed by Schwann cells. Okay, so the degeneration involves the phagocytosis of the dead parts of the cells. And then what happens is the living part of the cell that's still attached with the cell body and nucleus and so forth, well, it'll start to regenerate. And so growth factors are released that stimulate the growth of the axon, and these are released by remaining Schwann cells. And they essentially entice the what's called the growth cone, which is at the very tip of the regenerating axon, to then search out and finally get to the muscle. So they sort of lead the axon to the muscle. So they're very important in this sense. And eventually the new axon connects to the undamaged axon, the effector, which in this case is a skeletal muscle fiber. It could be, recall the word effector, could be smooth muscle fiber. It could be something like a gland also. One topic that's of great interest is CNS regeneration. As I just explained, the PNS can regenerate reasonably well. CNS regeneration is a little bit more problematic. So when you have injury of the CNS, this can stimulate growth of axon collaterals, but often the central axons have a more limited ability to regenerate than peripheral axons. So we believe that one protein that's involved in this is a protein called NOGO. There was improved axon regeneration in rodent models of spinal cord injury and stroke when animals were treated with antibodies that neutralized the action of NOGO. So in other words, this protein seems to, it's produced by oligodendrocytes, and it seems to inhibit regeneration of axons in the CNS. So blocking that protein seems to promote regeneration. So that might be useful in the future with therapeutics. There's also been a lot of interest in the potential use of stem cells for human spinal cord injuries. So, for example, there have been a number of studies using animal models. For example, you could take transplanted neural stem cells, you can inject them in the site, and then these will actually sprout thousands of axons. And they can actually have been shown to restore hind limb function in rats with a transected spinal cord. So it looks like there's definitely potential therapies and so forth out there in the future for this, for humans. Neurotropins are a family of proteins that induce survival, development, and function of neurons. So these are called growth factors. So the most famous of these is nerve growth factor, NGF. And there's another one called brain-derived neurotrophic factor that's also quite well known. It's also glial-derived neurotrophic factor, GDNF, and then neurotrophin 3 and 4/5. So these neurotropins are particularly important for growth in the fetal brain, but they've been shown to be very important also in the maintenance of synaptic sympathetic ganglia and the regeneration of sensory neurons in adults. Okay, there's a lot of research and interest in astrocytes because they're very important. They're the most abundant type of glial cell. As we mentioned earlier, astrocytes have many processes, hence the name astrocyte. Astra has to do with star shaped. They have end feet processes that are associated with capillaries within the brain. So they actually help to induce the endothelial cells of capillaries to form what are called tight junctions. Tight junctions are junctions that prevent paracellular movement of materials between cells. And so this sort of forces materials to go transcellular, that is, through the actual cells that make up the endothelium of the capillaries in the brain. And this essentially is the main part of the blood-brain barrier. This prevents substances from coming in easily into the brain. And this is, of course, important for medicine because there are also medicines that can't enter the brain very easily. And so we have to give alternative forms of medicines that can, in fact, pass through the blood-brain barrier. The astrocytes also, these feet also take up glucose from the blood in the capillaries. The glucose is then metabolized to lactic acid, and then you have lactate, which is the ionic form of lactic acid. And this is released at the foot processes on neurons, and it's taken up, and the glucose can then be used as a type of, essentially, it's oxidized to form ATP. There's another thing shown here also, which is there are feet processes on synaptic terminals or axon terminals of neurons. And so what happens is glutamate is released, that's a neurotransmitter, into what's called the synaptic cleft. It diffuses away from the synaptic cleft. It's picked up by these end feet processes of astrocytes. It's then converted into glutamine, and glutamine is then given back essentially to the neurons, which then convert it back to glutamate. Another well-known function of astrocytes is that they help to keep the environment stable for neurons. And one of the main functions they do is to take up potassium. Remember, we talked about how potassium is extremely important for excitable cells such as muscles and neurons. Recall that the main permeability at rest of these cells is potassium permeability, meaning that there's a large number of leak channels, potassium channels, that are open relative to the leak channels for sodium or chloride. So small changes in potassium extracellular concentration will lead to large changes in the resting membrane potential of both neurons as well as muscle cells. And so this could be quite damaging. So for example, if the neurons depolarize too much, they'll become too excited and they'll generate a huge trains of action potentials. They could potentially die. And in the other extreme, if the potassium levels are too low, then the resting membrane potential will become hyperpolarized relative to where it should be, and the neurons will become less active or less excitable. So in order to compensate for this, astrocytes will take up potassium from the extracellular environment, and this helps to maintain the ionic environment for the neurons so that they're happy and healthy. And we mentioned this already. So these end process will take up glutamate that's released from the synaptic clefts. As glutamate is released from the axon terminals, it's converted to glutamine, which is then released back to neurons, which convert it back to glutamate, so it can be released again. Neurons generally don't store a lot of glycogen. Muscle cells do. For example, skeletal muscle cells have a lot of glycogen stored in them. Neurons don't, but their very metabolically active because of all the processes that are doing. The release of neurotransmitters is an energy intensive process. Supporting action potentials is an energy intensive process. So they need energy constantly. And so the way the brain works is that the astrocytes can store glycogen. And then they can produce lactate, which is then essentially transferred to the neurons and can be used as an energy source. So the lactate is oxidized. So your book really goes, the author seems to really love astrocytes, and for good reason. These are very important glial cells. They're also needed for the formation of synapses in the CNS, as you can see here, and processes end feet of an astrocyte are actually right there at the axon terminals. So they're very important for synaptic function. They can also regulate neurogenesis in certain regions of the adult brain. Astrocytes, as I mentioned earlier, they induce the formation of the blood-brain barrier. So these end processes on the capillaries will actually release local chemicals that act very locally to stimulate the endothelial cells to form the tight junctions that make up the blood-brain barrier. And actually, astrocytes can actually release transmitter molecules. These are called gliotransmitters rather than neurotransmitters. And they can stimulate or inhibit neurons. And the types of molecules that we're talking about would be things like glutamate, adenosine triphosphate, or ATP, adenosine, and D-serine. I've already mentioned the blood-brain barrier, but this is quite important. One reason it's important is your brain is very sensitive, and so you wouldn't want, for example, bacteria getting into it. You wouldn't also want toxins getting into your brain. And one way of preventing bacteria and toxins from getting into the delicate brain tissue is the blood-brain barrier. So we have capillaries in the brain that are non-porous. In other words, adjacent cells are essentially tied together by what are called tight junctions, which involve proteins between one cell and the next. Now, this is a bit different than capillaries in other parts of the body. There are other capillaries that don't have tight junctions and actually allow for paracellular transport. So essentially, by having these tight junctions, it forces everything to move through the cells themselves that make up the endothelium of the capillaries. And so this allows the brain to be much more selective in terms of what enters it. So substances can only move through the endothelial cells by transcellular movement, so not paracellular. They move by simple diffusion. So gases, for example, like oxygen and CO2 can move very easily through membrane, and we talked about that in the past. But you also have, for polar molecules that are a bit larger than, say, glycerol or urea, you would need selective active transport processes. So our example, for example, would be glucose is a polar molecule. It's a bit larger than urea or glycerol, and it can only move through a cell membrane by active transport processes, or potentially, this isn't completely true here. It could also be through transport processes in which you have facilitated diffusion, where glucose, for example, is moving down its concentration gradient. So it could be active or it could be facilitative transport processes. So the blood-brain barrier is a good thing, but it can create problems when you're trying to get chemicals into the brain, like for chemotherapy. So the brain, basically, you have difficulty with penetration of the blood-brain barrier. And you can get around this in a couple different ways. One is you can make the blood-brain barrier temporarily more porous. And one way you can do that is to essentially inject the person through an IV solution. Specifically, you'd put it, actually, it would be an injection to arteries that are going specifically to the brain, or I suppose you could do an IV solution. But you would put in something that would be hypertonic, which would cause cells to shrink a bit. And that'll actually break up the tight junctions a bit, just enough to get the drug in. The other possibility is to simply use forms of a drug that can pass through the blood-brain barrier, but then are altered and metabolized within the brain, and then are active within the brain. So this is a picture of a normal blood cell, blood vessel, where many of them are quite porous. So you have these waterline pores. This would allow for many substances to pass through quite easily. Um, Here's a picture of the blood-brain barrier. You have tight junctions, so nothing is allowed to pass through paracellular between the cells. And you also have these glial brain cells that are helping to support the blood-brain barrier. The next topic is electrical activity and recall that neurons have a resting membrane potential of about negative 70 millivolts. Um, and and we've talked a lot about how that's established, right? There are a couple different things. One is that there are large negative molecules inside of cells, and we really didn't go into this in great detail. You could look it up if you want to. Just it's not going to appear on a test or anything, but there's the Gibbs-Donin equilibrium, which basically tells us that if we have large negative molecules that are inside of a cell, and these molecules are non-penetrating, in other words, they're sort of stuck within the cell, then they can lead to a negative potential inside of the cell relative to the outside of the cell. The other thing we talked about in detail before was the sodium-potassium pump. So the main thing that this pump does, is to establish ionic gradients for sodium-potassium. But recall, and that's the main force, but that leads to a resting membrane potential in -70, having these ionic gradients. But the other thing that's actually less important, but also contributes, is the fact that the sodium-potassium pump is electrogenic. So because the pump will pump 3 sodium out for 2 potassium in, with each cycle of the pump, you essentially have positive charge moving out and leaving behind a negative charge. And so this is also going to make the cell slightly negative. And finally, we talked about this before. We talked about the Nernst potential. So the permeability of the membrane to charged ions is different. At rest, a cell has a large potassium permeability. The chloride permeability is typically a little bit less than half of that of the potassium permeability. And then finally, the sodium permeability is much less than that of potassium. So because of this, because the permeability to potassium is the highest, potassium has the largest effect on the resting membrane potential. Recall that the equilibrium potential for potassium is about -90 in your typical cell. And so that's quite close to the resting membrane potential of the cell. In a typical neuron, the equilibrium potential for sodium, we've talked about this before, is about positive 55 to plus 60 millivolts, somewhere in that range. And you'll note that that's very different than the resting membrane potential. The chloride equilibrium potential is actually quite close to the resting membrane potential. It's either equal to it, around -70, or it might be slightly more negative in a typical cell. But the chloride permeability is lower than that of the potassium permeability, therefore it has less of an effect on the resting membrane potential. Now recall that at rest, there's a high concentration of potassium inside of a cell, and the opposite is true for sodium. There's a high concentration of sodium outside of the cell. Okay, recall that neurons and muscle cells are excitable, meaning that they can generate action potentials, which are also called spikes, or sometimes called nerve impulses. The basic idea is that they're able to change their membrane potentials very quickly. So these changes in what causes this change in membrane potential? Well, there's very large changes in the permeability to certain ions. So for example, there's a net movement of ion. The net movement of ion is dependent on its electrochemical gradient. So this is a combination of a chemical gradient and electrical gradient. So let's talk about some ions. So for example, potassium is positive. There's a higher concentration of potassium within a cell than outside of the cell. So it would have, if you just looked at this chemical gradient, potassium would tend to flow outside, from inside to outside. However, we mentioned the fact that the cells are negative inside of its we have a net negativity inside of a cell relative to the outside of the cell. Therefore, the electrical gradient for potassium is actually in the opposite direction. The inside, there's an electrical field across the cell membrane such that it's negative inside with respect to outside. Potassium ions are positive, so that would actually tend to hold potassium ions in. And those two things can be balanced. Remember, this is the idea of the equilibrium potential. That exactly the equilibrium potential for potassium, EK, which is negative 90 millivolts in most neurons, then the tendency for potassium to leave the cell due to its chemical gradient is exactly balanced by the tendency for potassium to enter the cell due to its electrical gradient. So potassium, in a sense, is happiest. It's kind of dangerous to anthropomorphize ions, but it's happiest if the membrane potential would be negative 90 millivolts. Sodium's a bit different. Remember, recall that sodium is higher in concentration outside. So its chemical gradient would tend to, for a cell that's at rest, would tend to force sodium to go down its concentration gradient and enter the cell. Now, at rest, a neuron is also negative inside with respect to outside. Therefore, the electrical gradient is in the same direction. So there's quite a strong electrical chemical gradient for sodium that favors the influx of sodium. So there's a net influx of sodium into the cell. And we'll see that is very important during an action potential. There'll be a large influx of sodium into the cell during an action potential. So flows of ions are called ion currents, and these occur only through ion channels. And we've discussed why this was in the past. We've talked about the fact that ions are charged, whether they're negative or positive, and they have a hydration cell, which is essentially they have a group of water around them. So they're essentially hydrophilic. Ions associate very well with water. They do not, however, associate very well with hydrophobic tails of the phospholipids within the membrane of a cell. And so they really can't enter a cell very easily by simple diffusion through a cell membrane. They have to enter through channels. Okay, we've actually talked about this a bit in class already, but the idea is that, let me see if I can draw it. We have a cell, and here is its, I'm drawing its resting membrane potential. And if the resting membrane potential were to go up, that would be a depolarization. If it were to go down, that would be called a hyperpolarization. Okay. So let's see. We have these terms. So the neuron is considered to be polarized because it's negative, right? Negative 70 millivolts with respect to outside. When the membrane potential inside the cell increases and becomes more positive, this is called depolarization because it's becoming less polarized, right? It's going towards zero. And technically, when you go above zero, which does happen during an action potential, well, it's being polarized in the opposite direction. But we tend to just use the term depolarized even in that. And we just say, well, anything in the upward direction will be depolarized. And then as we, as the, say the membrane potential returns back to its normal value, we can also use the term repolarization. We're bringing it back to its normal resting value. When the membrane potential inside the cell decreases or becomes more negative, this is called hyperpolarization. So we could call this both hyperpolarization because it's going downwards. We can also refer to this as repolarization because this neuron started at a certain resting membrane potential, it depolarized, and then it repolarized back to its original value. This is just a larger version of the same picture I showed a second ago, but I just wanted to point out that remember, when we record these membrane potentials, we're recording using two electrodes. One is placed inside of the cell, it's an intracellular uh, recording. And then the other electrode, which is sort of the reference electrode, is just sitting outside the fluid, outside of the cell. And so when we talk about membrane potentials and things like that, we're talking about the difference between the inside and outside. And by default, definition, we sort of set the extracellular fluid potential to be 0. And so the inside happens to be negative with respect to that. And so you always have a negative value for resting membrane potential. Okay, let's talk a little bit about how you could get a depolarization. Well, one way you could do it is to have positive ions enter the cell. So for example, sodium might enter the cell. And you could do this a couple different ways. One is you could do it experimentally. You could inject positive ions such as sodium into a cell. Positive ions going into the cell will make the inside less polarized, or it would depolarize. You can go in the other direction. Hyperpolarization occurs when positive ions leave. So for example, if you have, typically, remember, we have a lot of potassium ions inside. And so you could have potassium ions leave. As they leave, that would leave behind negative ions. And so the cell would become hyperpolarized. The other possibility is you can have negative ions enter the cell, such as chloride, and that would tend to also hyperpolarize the cell. And again, these are things you could do experimentally. It'd be quite easy to, say, inject positive ions such as potassium. Well, sorry, you couldn't do that easily. You could inject chloride ions, which are negative, into the cell, and that would hyperpolarize the neuron. Potassium is already high inside, so injecting potassium ions into the cell would have a little more subtle of an effect. Okay, depolarization of a cell is considered to be excitatory. Why is it considered to be excitatory? Well, we haven't gotten to this topic yet, but it'll make the cell more likely to generate what's called an action potential or impulse or spike. And hyperpolarization is inhibitory. So if you hyperpolarize a neuron, then it'll be less likely to generate an action potential or spike. Okay, just to be clear, because I think I might have confused people a little bit. If you were to say inject sodium into a cell experimentally, that would depolarize the cell, right? That would make sense. Potassium ions are also positive. So if you were to inject them directly into the cell, then that would also have a depolarizing effect. Okay, so there's different types of channels. So we've talked in the past about leak channels. We talked about potassium leak channels, sodium leak channels, and chloride leak channels. These are channels that are open all the time. In addition to those sorts of channels, there are also voltage-gated channels. Voltage-gated channels will open when the membrane potential changes. So for example, voltage-gated potassium channels open when a particular membrane potential is reached. That's not exactly true. It's not exactly one particular membrane potential, but as the membrane potential becomes more depolarized, more and more of these voltage-gated potassium channels open. And the reason for this is each one of them responds similarly, but there's a little bit of randomness in how they respond. So as the membrane potential becomes depolarized, more and more of these channels will open. And that process of opening is called activation. Then let's imagine, let me draw this so we can get a little bit of a handle on what this looks like. So we'll draw some membrane. It's a terrible drawing. Here's the channel. So the channel has a pore in the center. And so if this is let me see if I can draw better here. If this is the outside and this is the inside, so these potassium channels would open in response to depolarization. And we've already established that then the potassium would tend to flow out through the channel. and that will tend to hyperpolarize the neuron, bring it. So if it was depolarized, this would tend to oppose the depolarization. Now, this channel is actually closed most of the time. So let me see if I can draw what that would look like. So channel is sort of closed and doesn't allow potassium to pass through. And then it can open, allowing for potassium to pass through. All right. So a voltage-gated potassium channel has one gate, and the gate happens to be located essentially more or less in the center here of the protein. So this protein, of course, it's if you look down on it, it'd be shaped like a donut, right? It has a hole essentially through it that allows for potassium to move through, together with associated water in the hydration shell. Okay, so that's actually fairly easy to understand. So let's again go through this. So we do with the depolarization. Let me see if I can draw again. With the depolarization of the membrane, which could be caused by various things. For example, you might inject positive ions into the cell. Then the potassium channel will open, and that would tend to bring the membrane potential back down again. Okay. Now, sodium channels are a little more complicated. These are voltage-gated channels that are closed at rest, and they will open or activate in response to depolarization. So again, we could draw a channel. Sodium channel is looking something like this. So normally it's closed. And apologies for the bad drawing. And then with depolarization, the channel will open. And sodium, let's recall what we discussed earlier. Sodium has a higher concentration outside compared to inside. Sodium is positive. Then at rest, the cell is negative inside with respect to outside. So sodium would tend to flow into the cell down both its chemical as well as electrical gradient. So what would be the effect of this? Well, if you had a bit of a depolarization, well, sodium channels lead to, when they open in response to that depolarization, they're going to lead to influx of sodium, which would cause even more depolarization. And then as that depolarization will in fact activate or open even more sodium channels, causing more sodium influx. And so you get this sort of feedback in which you're now starting off at the resting membrane potential, you're now deviating very strongly in the positive direction or depolarizing direction from that resting membrane potential. So this would be an example of positive feedback mechanism or positive feedback loop in which you're deviating very far from the normal resting membrane potential. With potassium channels, it's, as we described a second ago, it's a bit different. If you have a depolarization, say caused by injection of current by an experimenter, well, the potassium channels will open, potassium ions will tend to flow out, positive ions moving out will leave behind negative ions, and so you'll tend to hyperpolarize and bring the membrane back down again. So that would be a negative feedback mechanism or negative feedback loop. Now, if this wasn't complicated enough, there's one more layer of complication, which is that these voltage-gated sodium channels also have a second gate, and that's called an inactivation gate. So let me draw it now with the inactivation gate. So when the cell is at rest, the activation gate is closed. You can see that what I've tried to draw here is a closed gate. And there's an inactivation gate, which is on the cytoplasmic side, right, on the inside of the cell. It's sometimes represented as sort of a ball and chain. So this is a bit of protein that hangs down intracellularly. Now, when the cell is at rest, that second gate, so this ball and chain can essentially move and clog the pore, essentially. So it can block it, right? So a channel is going to be closed whether the original gate, the activation gate, is open, is closed, then then this channel will be closed. It could be closed in a second way, which is that gate could actually be open, but if this inactivation gate moves into place, it'll also block the channel and make it closed. So sodium channels are definitely a little more complex. So what happens? Well, we would start off with a resting membrane potential with a cell. Whoops, let's go back to drawing. Looking like this, with this inactivation gate being open. Now, initially, the activation gate opens very rapidly. So if you have a depolarization, as I drew earlier, sodium would flux through, but then slowly the inactivation gate would start to move, just, a bit slower. So let me go back to drawing this. So that's meant to look at like it's open, okay? So the activation gate would still be open, and yet we now have the inactivation gate closing the channel, okay? And so this allows for sodium to flux through the open channel earlier on because the activation gate is open. We should have the inactivation gate. But the inactivation gate is still open. I can't draw it very well. So it hasn't moved yet. And then eventually, the activation gate may still be open, which isn't drawn very well here, but that's supposed to be open. And then the activation gate comes into place. So this bit of protein flops into place. The ball of the ball and chain then blocks the channel. And then the channel is effectively closed at that point, because if either of the gates is closed, then the channel is closed. Okay, so keep all this in mind, and we'll come back and talk a little bit more about these in the next few slides. Okay, so we've already begun to talk about these voltage-gated sodium channels. These channels open if the membrane potential depolarizes to about -55 millivolts. Now, your book likes to make it sound as if these things are just on and off, but it's not really the case. As the membrane potential becomes more depolarized, more and more of these sodium channels will open or activate. But this is true around -55 millivolts. That's about the level in which you have spike threshold. So if a neuron reaches about that level of depolarization, remember it starts off at -70 millivolts, then you'll often generate a spike or action potential. So we've already discussed this. Sodium rushes in due to the electrochemical gradient. The membrane potential climbs towards the sodium equilibrium potential, which recall is around +60 millivolts, more or less. These channels are slowly inactivated in response to depolarization, and that's due to that inactivation gate that I just talked about. So what this allows for is that sodium will rush in, but it won't continue to rush in. There'll be an endpoint which the inactivation gate closes, and so the channel will effectively closed, and then that stops. That process stops. So here's an artist's rendition of what I've been talking about. So of course, it's much more beautiful than what I was able to draw. Here's the channel when it's closed. So that we can assume here the cell is at resting membrane potential. So the activation gate, which is this one here, is closed, but the inactivation gate, which is represented here by this protein that looks like a ball and chain, is open. It's not blocking the channel. This activation gate, which is towards the center of the channel, responds very quickly when the cell depolarizes and it opens. This allows for the flux of sodium down its concentration gradient from outside to inside. And then with a little bit of a delay, the inactivation gate does in fact respond to this depolarization by moving. It says there's a shape change. It now blocks the channel, and then this channel is effectively closed. And then eventually what they're showing you here is this final arrow is eventually if the channel has been returned, if the membrane potential has been returned back to the resting membrane potential for a length of time, then the activation gate will close. So it closes up in response to the fact that the cell's no longer depolarized. It's at the resting membrane potential. And then slowly the inactivation gate opens. So the inactivation gate is essentially just slow. It was slow to close the channel. And then even once you've returned back to the resting membrane potential, it's relatively slow to open again. So this effectively means that even after an action potential has occurred, or towards the trailing end of an action potential, the sodium channels will be closed. Okay, we've already talked about some of these points, but it's worth going through them again just to make sure, because this is relatively complex. In response to depolarization, the voltage-gated potassium channels open. Potassium rushes out of the cell, so this would tend to hyperpolarize the cell. This makes the cell repolarize back toward the potassium equilibrium potential, which is negative 90 millivolts. Voltage-gated potassium channels are slower to open or activate compared to voltage-gated sodium channels. So this is a very important point. If both voltage-gated potassium channels and sodium channels, they're both activated or open in response to depolarization. Now, if they both opened at the same speed, then the action potential wouldn't occur because the sodium would rush in, potassium would rush out, and there wouldn't be any action potential. So in order for an action potential to occur, you need for the voltage-gated sodium channels to activate or open very quickly. And we need for the voltage-gated potassium channels to open quite a bit slower. So they're both activated or opened by depolarization, but the voltage-gated potassium channels are slower. And so this allows for the membrane potential to move upwards towards the equilibrium potential for sodium. At the top of an action potential at the end of the rising phase, you're typically at about plus 30 millivolts. So you're not at plus 60 millivolts, which is the equilibrium potential for sodium, but you're getting close to it. And then due to two processes, the inactivation of the sodium channels, meaning that they close, and also the slower opening of the voltage-gated potassium channels in response to depolarization. Then as the potassium rushes out of the cell will repolarize. Okay, let's go through this another time. I think we talked about it already, but we're starting off with a cell being at negative 70 millivolts. For some reason, say we injected current, we reach the threshold membrane potential of negative 55 millivolts. Voltage-gated sodium channels open, sodium rushes in. This causes the cell to depolarize even more. This causes more sodium channels to open, and the cell becomes more and more permeable to sodium. So one way of looking at this is that due to the voltage-gated sodium channels opening, there's an increase in permeability to sodium. So recall that we talked about this earlier. If our cell was permeable exclusively to sodium, then the resting membrane potential of the cell would in fact be the equilibrium potential, which would be about positive 60 millivolts. Now, it turns out we never quite get there because, of course, it's also permeable to other ions. There are leak channels for potassium, leak channels for chloride. That prevents the membrane potential from reaching that plus 60. And also the fact that we have sodium channel inactivation, that also means the sodium channels will start to close. And additionally, the other reason we don't reach the positive 60 millivolts is because we have the opening of voltage-gated potassium channels, although they're slower than the voltage-gated sodium channels. So recall again that this was an example of positive feedback. So we have this depolarization that's quite dramatic, going up to about plus 60 millivolts. So that's the rising phase. Then you have the falling phase of an action potential. It drops down, and then it returns eventually to the resting membrane potential. Note that there is a period of in which the resting membrane potential actually goes below the membrane potential actually goes below the resting membrane potential. So we have a couple different terms here. One is the fact that this resting membrane potential goes above 0 millivolts. So that's called the overshoot. And then when it goes below the resting membrane potential, that's referred to sometimes as the undershoot, sometimes it's referred to as the after hyperpolarization. Again, we've talked about this already, but it's probably worth just going through this again. So again, we have the rising phase of the action potential, which is quite dramatic due to sodium rushing in. The sodium channels inactivate, and then the potassium channels open. And so due to this, you have the repolarization of the membrane during the falling phase of the action potential. And then it finally returns back to the resting membrane potential. So the activation of the potassium channels can be considered to be a negative feedback loop. It brings the membrane potential back towards the resting membrane potential. It actually undershoots, as I mentioned earlier, a little bit, but that's relatively small. And so we would generally consider this to be a negative feedback loop. Okay, so this picture essentially just is from your book and essentially goes through all the things we just discussed. We start with a depolarizing stimulus. Voltage-gated sodium channels are opened. Sodium diffuses into the cell, so there's a net influx of sodium. Positive ions moving into the cell causes depolarization. This has a positive effect, in other words, on the voltage-gated sodium channels. In other words, it causes them to activate or open. That causes more sodium to diffuse into the cell, which causes more depolarization. So this would be considered a positive feedback loop. Now, at the same time, the same initial depolarization stimulus, together with the depolarization caused by the sodium channels, causes voltage-gated potassium channels to open. Potassium will flux out of the cell, so there'll be a net diffusion of potassium from inside to outside. So positive ions are leaving, they're leaving behind negative ions within the cell, and this will tend to hyperpolarize the membrane or bring it back. So again, that's a negative feedback loop. And here we can see a picture of all this. We have initial stimulus shown here in pink. We have then sodium fluxing in. We reach the top of the action potential around plus 30 millivolts. Potassium then fluxes out, and you go back down and actually go below the original resting membrane potential. This is another figure from your book, and it gives some good insight into what's happening. First of all, this top figure shows the membrane potential, right, versus time. So here's the membrane potential going up and then down, and then finally returning to the resting membrane potentials. Time is on the x-axis. Now, another way of looking at this is you can look at the movement of sodium and potassium. They're calling it sodium-potassium diffusion. Technically, what we're really seeing here is the sodium and potassium permeability. So that would be a better way of indicating this, because this is actually a little bit incorrect for technical reasons I don't really want to get into. If you're actually showing the flux of sodium and potassium, the graphs would look a bit different. So what's really being shown in the y-axis is the sodium and potassium permeability. So let's think about this. So you start off at time 0, you have some sort of initial stimulus, the sodium channels open, and so you would have expected there'd be a normal, a little bit of sodium channel permeability, right? It's not shown here, but it's relatively small. That's to the sodium leak channels. Then you have the voltage-gated sodium channels opening, so the sodium permeability would go up very high. And then remember, there are a couple processes going on here. There's the sodium channel permeability will decrease because the voltage-gated sodium channels are closing due to the inactivation gate closing. And also, as you go back down, the activation gate of the sodium channel will actually close also as you get down towards the lower levels here. So there are really two processes, but the sodium channel inactivation is probably the main one that's really causing this permeability to go down, at least initially. Now, we talked about the potassium channel also being activated or opened in response to depolarization, and that's absolutely true. So again, there's normally a bit of resting, quite a bit of resting per, permeability to potassium through leak potassium channels. That's not shown here. What we're just looking at here is the voltage-gated permeability through voltage-gated potassium channels. So that increases in response to depolarization, but it occurs with a bit of a delay because these channels are slow. So these go up. Now, remember, so this permeability goes up, and that actually leads to potassium then fluxing out of the cell, which happens to bring the voltage back down again. And so remember, these voltage-gated potassium channels are activated or opened in response to depolarization. Here, as in the falling phase of the action potential, the membrane potential is coming back down. And so these channels will actually naturally start to close because they're not being stimulated as much. They're not being activated or opened in response to depolarization. But note that they're kind of slow to close. So this is another key point. You see how the sodium channels activate and then inactivate relatively quickly. Potassium channels activate and then deactivate is the correct technical term as they respond to the hyperpolarization of the cell. But they're still open for a considerable amount of time, about a millisecond or so after the peak of the action potential. Could be even more than that, quite a few milliseconds. And so what happens is you now have an excess of potassium permeability compared to the initial resting membrane potassium permeability. Remember, at rest, there are leak channels for potassium that are open, but the voltage-gated potassium channels are pretty much all closed. Immediately after an action potential, you have the leak potassium channels. And additionally, you have now the voltage-gated potassium channels that have been opened or activated, but they're slow to close. So they're open, and that means you have a higher membrane permeability to potassium than you would have normally. And this tends to bring the membrane potential closer to the equilibrium potential for potassium, which is negative 90 millivolts. Okay, we've largely already covered this topic. This after hyperpolarization is also sometimes referred to as an undershoot. So when the cell repolarizes, the membrane potential undershoots the resting membrane potential and gets down to about negative 85 millivolts. So it's getting close to EK, the equilibrium potential potassium, but never quite reaches it. Well, why doesn't it reach it? Well, because there are still other permeabilities. There's leak channels for sodium and leak channels for chloride. And both of those have equilibrium potentials that are depolarized relative to the equilibrium potential for potassium. So you'll never quite reach negative 90 millivolts, no matter how many voltage-gated potassium channels you have open. It doesn't reach the potassium equilibrium potential because voltage-gated potassium channels are inactivated. And this is completely incorrect, actually. So your book refers to this as inactivation of voltage-gated potassium channels, but this is not correct. What happens is that voltage-gated potassium channels are activated or open. And then as they close, as a natural response to the fact that the membrane potential is coming back down again during the falling phase of the action potential, that process is called deactivation. It's not inactivation. The term inactivation is reserved for channels such as the sodium channel that have a specific inactivation gate, which we just talked about a few minutes ago. So this is technically quite incorrect, okay? And also, so the other reason you don't get to EK, or equilibrium potential for potassium, is that, as I mentioned, there are also sodium leak channels that are open. And those would tend to drive the membrane towards plus 60 millivolts. Now, those are not a lot of them, right? So they're sort of overwhelmed by the potassium channels, but they do have a slight influence. Finally, going on in the background, so the sodium-potassium pump really has negligible effect on the actual shape of an action potential because it's just a very slow process, where an action potential is a very fast process. But we should remember that it's sort of like the hero and behind the scenes. It's maintaining the resting membrane potential by maintaining the potassium-sodium gradients at all times. Action potentials are referred to as all or none events. Once threshold has been reached, an action potential will happen. It's very difficult to stop it from happening. The size of the stimulus doesn't really affect the size of an action potential. So as long as you could get to, for example, if you get to the negative 55 millivolts really quickly versus slowly, it doesn't really have any effect. The action potential will be about the same size. It will reach about positive 30 millivolts. Therefore, the size of the stimulus does not affect the action potential amplitude. The amplitude is, from its peak to its base during the after hyperpolarization. The size of stimulus also doesn't affect the action potential duration. Now, a quick aside, for all intents and purposes for this course, we'll just call these action potentials as having all the same size and shape. There are slight variations. There are situations where you might have a bit of sodium channel inactivation. If you just had an action potential, for example, just prior to another action potential, you might still have some sodium channels that are inactivated, and therefore you might see a slight reduction in the peak of the action potential. Okay, but those are minor effects. Okay, this is just an illustration from your book showing the point that we can have a weak stimulus, and it's not even shown. You can't even see the stimulus, and so there's no action potential. And then they're showing a series of depolarizing shocks given to the axon of, say, a neuron, and you'll always get action potentials that are essentially identical. They all have peaks that are about the same size. So a question you might have is, well, if action potentials are all the same, how does your body know, for example, if something presses down on your finger lightly versus a much stronger stimulus? Well, there's a couple ways you would code for that sensory stimulus. And one way is that the individual neurons would generate different numbers of action potentials or different frequencies of action potentials. So for example, here we have a stimulus. It's causing some sort of depolarization of the neuron, which isn't shown here, but you can imagine it happening. Now you have a stronger stimulus, and now you have a greater depolarization, and you have a certain frequency of action potentials. Now, finally, in this third case, you have an even stronger stimulus. Now note the action potentials are all identical. They have the same amplitude, but there's a higher frequency in the action potentials. And so this would tell us that, oh, there's actually something pushing down a little harder on our finger. Now, in addition to this, in terms of coding sensory stimuli, as you push down, say, with a little more force on your index finger or something like that, not only would individual neurons be undergoing this process, what's called, coding for frequency, coding the stimulus strength through frequency, there would actually be an increase in the number of sensory neurons that are activated as sensory neurons in neighboring regions of the skin would also start to pick up the stimulus and so forth. So that's referred to as population coding. You'd have more neurons active, but you'd also have a higher frequency of action potentials within the neurons that are active. Turns out there's an upper limit to how fast a train of action potentials can be. In other words, what the highest frequency can be. And that's because immediately after an action potential, there are essentially two periods called refractory periods. One period that's called the absolute refractory period is a period in which no matter how much strong a stimulus is, you can't possibly get another action potential. This is due primarily to the fact that the sodium channels are inactivated. So recall that initially the activation gate of the sodium channel is open, so those open, sodium rushes in, but then due to the depolarization, the inactivation gate closes the sodium channel. So once they're closed, you can't really get an initial action potential, say, right away. And the inactivation gates, recall, are relatively slow. So once they close the channel, they stay closed until the membrane has actually had some time to recover by being hyperpolarized back down near the resting membrane potential. So that's the absolute refractory period. There's another refractory period called the relative refractory period. And this one's primarily due to the fact that, well, it's actually a little complicated. It has the sodium channels may still be inactivated. to some degree. Now remember that there's not one sodium channel. There's thousands and thousands of them. So, and they all respond identically. They're all identical proteins, but they respond in a random way. So some sodium channels may essentially that inactivation gate may move back and open up, whereas other may still be inactivated or closed due to the inactivation gate being closed. And so because of this being a population of channels, it will take some time for them to recover. But during that period, once you've had the recovery of enough of these sodium channels, you can in fact get another spike if you give a strong enough stimulus during the relative refractory period. The other thing that contributes to the relative refractory period is recall that there's potassium channels that have opened, and remember they were slower to open than compared to sodium channels, and they're also slow to close, and so they tend to be open and they tend to hyperpolarize the membrane a bit below the resting membrane potential. So that also definitely contributes to the relative refractory period. Now, during this relative refractory period, you can overcome both of these things: the fact that some of the sodium channels are still activated, and the fact that there are voltage-gated potassium channels that are still open. If you give a strong enough stimulus, you can in fact get another action potential within the relative refractory period. So we've already discussed this about the potassium channels. And so another point is that each action potential remains a separate all or none event. The action potentials never fuse one into another. They always stay separate events. Okay, we've talked about things like action potentials, but we haven't really talked much about what happens within a neuron with essentially sub-threshold events. You have events such as, for example, if you have the dendrites of a neuron and there's a stimulus and there's a depolarization of a dendrite due to, say, sodium fluxing into the dendrite, well, that depolarization will actually spread throughout the neuron, and it may in fact trigger an action potential near the axonal hillock and initial segment of a neuron. And whether it does or not depends a lot on various factors. And one thing is the cable properties of a neuron. So this term cable properties comes from the idea that you can study neurons with a lot of the same equations that you would apply to, say, underwater cables or electrical lines or telephone cables and so forth. Now, the properties are quite different. If you think of like a copper wire or something like that, then there's a relatively low resistance to the movement of electrons longitudinally along the length of the copper cable. And typically you have rubber or some sort of insulator around the copper, which is the conductor. And so there's very little leakage of electrons out. Turns out that neurons are very much different, even though the same equations apply, the actual values are very different. So neurons have some ability to conduct charges through their cytoplasm. So remember that within the cell, you have a number of charged ions, sodium, potassium, chloride, things like that. So those allow for the movement of charges. But there's a fairly high internal resistance to the spread of charges. And the other thing is that these neurons are actually fairly leaky. So these charges, which are sodium, potassium, chloride, principally also calcium to some degree, they can leak across the membrane through these leak channels that we've discussed. And so a neuron can't really depend on cable properties, so just allowing for charges to move down lengthwise along the axon to communicate from, say, one end of a long axon to the other. They're going to rely on action potentials instead. But it's necessary that we understand these cable properties because it helps us understand how neurons are able to integrate information at the level of the dendrites. And it also tells us something about how things like the conduction speed of an action potential. Okay, here's a picture from your book showing a stimulus. So we're injecting positive charges causing a depolarization with a stimulating electrode. So that's occurred here, where this positive plus sign is here. Now, these charges actually go in both directions, but we're just showing you in one direction along this axon. So the charges, if you inject positive charges, they're going to tend to be repulsed from one another, so they'll tend to move down the axon. Now the membrane, let's say this is sodium. The membrane does have a resting permeability to sodium, so some of the sodium will leak out across the membrane. And so basically what will happen is if you record at different points along the stimulus is the largest where you initially, in the local area we initially injected the current. If you record further down, you see it's a bit smaller, so it's a decrement or attenuation of the signal. A bit further on, it attenuates even further, and a bit further along, it's practically reduced to zero because, again, there's a bit, depends on two things. One is the ease with which the ions can flow longitudinally, and also the leakiness of the membrane itself. Okay, as I mentioned a second ago, these cable properties are very helpful for trying to understand how an action potential actually moves. So recall that we talked about the fact that an action potential might be generated around the area of the axon hillock and the initial segment of an axon. Now, if it just stayed there, it wouldn't really do much good, right? The action potential has to move along the axon all the way to the axon terminals, and each place the axon bifurcates into collaterals. It should continue to move along those collaterals also. So how does it occur? Well, recall that during the peak of an action potential, you have a lot of sodium flux into the cell. Now that sodium will then move actually in both directions from that site where there's influx. And as it does that, what it does is it'll depolarize the adjacent patch or piece of membrane. And so typically what will happen is that will then bring the next piece of membrane to a spike threshold. And then a new spike is generated at that point. And then again, there's a large influx of sodium. And then this depolarizes the next patch or piece of membrane. And then that will generate a full-blown action potential. And so the action potential can essentially move, it's a wave that moves down along the length of the axon. And if you did this, so for example, right in the center of an axon, so you injected current, then the action potential would actually flow in both directions toward the axon terminals, and it would actually flow backwards towards the cell body. But since these action potentials are generally generated near, for example, in this neuron is being generated near the cell body, there's a much lower density of sodium, voltage-gated sodium and potassium channels within the cell body itself. In fact, it's very low. So the action potential doesn't really invade the soma as much as moving down the axon in this direction. So this whole process can be thought of as a wave, right? And so, you've seen this, phenomenon where people, generate a wave within a stadium. And so this is a very similar process in which you have a group that's local in one area. They stand up, they wave their hands, they yell, and that stimulates the next group to do the same thing, which stimulates the next group to do the same thing. And so you have a wave that moves across the stadium. Now, the people that had just done it, well, they're now, tired of doing it, so they're sitting down. And so something very similar occurs within the axon of a neuron. The region in which an action potential had just passed, the membrane is in a refractory period, as we talked about it, right? There's an absolute and a relative refractory period. And largely that's due to the fact that the sodium channels, the voltage-gated sodium channels are inactivated. And so they can't generate another action potential right away. And so that's very important because it means that an action potential will travel in one direction away from the initial stimulus site. It won't double back on itself, in other words. So it turns out you have both myelinated and unmyelinated neurons in your body. Action potentials are produced in an unmyelinated neuron, action potentials are produced at every patch of membrane, at every point down the entire length of the axon, in the way we just described. And this means that in an unmyelinated neuron, the conduction rate's relatively slow because the opening of channels and the diffusion of ions takes time. The amplitude of each action potential is the same. It's conducted without decrement. Okay, in contrast to unmyelinated neurons, conduction in myelinated neurons is a bit different. I've already shown you pictures of what myelin looks like and the fact that there are these nodes of Ranvier. So recall that myelin is a type of insulation. And so what it'll do is it'll change the cable properties of a neuron. That wrapping of myelin, which recall is a lot of phospholipid bilayer, it's very fatty. It prevents the flow of ions such as sodium and potassium across the membrane. And so that essentially makes the membrane less leaky. And that means that any sodium that enters when an action potential occurs at a node of Ranvier, as the sodium flows in, then it'll flow a longer distance without decrement down longitudinally along the length of the axon until it hits the next node of Ranvier, which it would then depolarize if it hits spike threshold, and then you get influx of sodium and a full-blown action potential occurring at the next node of Ranvier. So in this way, the action potential actually jumps from one node to the next. It doesn't occur during the areas of the axon in which you have the myelin sheath. And again, another factor is that at these nodes of Ranvier, you have very, very high concentrations of sodium-potassium ion channels there. So this is referred to as saltatory conduction. Saltatory simply means jumping, and it's much faster than the type of conduction that occurs without myelin. So here's a picture of what we were just discussing, in which we have myelin wrapping around the sheath. You have the nodes of Ranvier. An action potential just occurred here. Sodium flux into the neuron, moves to the next node of Ranvier, depolarizing the membrane to the spike threshold. The second action potential occurs here. Sodium again fluxes in, moves down the axon to the next node, and you get an action potential at the next node. And recall, because of what we talked about, this action potential in this case moved in this direction because an action potential occurred here. And although you get an action potential at this next site of depolarization, blue shown here, these channels, sodium channels, are now inactivated. And so the action potential will tend to move in this direction, even though the sodium, when it fluxes in, will actually move equally in both directions. Okay, so action potential conduction speed is extremely important. We have different neurons that need to conduct at different speeds. So how can you increase the conduction speed? Well, we just talked about myelination. That can tremendously increase the conduction speed because the whole process, as I said earlier, of generating action potentials is relatively slow. Channels have to open, ions have to diffuse. But with saltatory conduction, those slower processes only occur at the nodes of Ranvier. The actual movement of sodium ions down the length of the axon is actually quite quick because they simply displace one another as they move along the axon. Another way of increasing action potential conduction is to increase the diameter of the neuron. This reduces the resistance to the spread of charges along the length of the axon. So that is actually sort of similar to, say, a copper wire. The larger the diameter, the lower the resistance to the movement of electrons down a copper cable. So some examples might be a thin unmyelinated neuron might have a speed of only 1 meter per second, which is relatively slow. A large diameter myelinated neuron might have a much higher conduction velocity of, say, 100 meters per second. So I'm going to end this video on this table from your book. It just shows, gives you some idea of why you might have different conduction velocities. So, for example, you have sensory neurons that detect muscle position. They might have a very large conduction velocity of 70 to 120 meters per second. Somatic motor fibers, these are motor fibers that go to skeletal muscle. They'll be relatively fast, 30 to 90 meters per second. Sensory neurons that respond to touch and pressure might have a conduction velocity of 15 to 40 meters per second. Sensory neurons that respond to pain and temperature might be 12 to 30 meters per second. The slower would be autonomic fibers moving to ganglia. So remember, the autonomic nervous system is involved in a lot of involuntary processes such as sweating, uh, movement of food through the GI tract, things like that. They don't have to be. These processes don't have to be that fast. So they might have a conduction velocity of only 3 to 15 meters per second. And then here we have autonomic fibers going to smooth muscle. So this could be things again like the GI tract, cardiac muscle, which of course through the muscles in your heart. And those can be relatively slow, 0.7 to 2.2 meters per second.